Modern artificial neural networks learn through an algorithm called backpropagation. The algorithm calculates errors at the output layer and mathematically propagates those corrections backward through the entire system. But biological neurons do not operate in reverse. In a physical brain, electrical impulses move strictly forward across synapses in sparse, one-way patterns. Backpropagation requires a computational concept known as weight transport. To accurately calculate how much a specific connection contributed to an error, artificial neurons must share perfectly symmetric synaptic weights in both the forward and backward directions. Biological synapses are inherently asymmetric. When a presynaptic neuron fires into a postsynaptic neuron, the chemical and electrical channels establish a specific directional strength. There is no physical infrastructure in human tissue to transport an exact symmetric weight value backward across that gap. Backpropagation also relies on global error computation. For any artificial hidden layer to adjust its weights, it requires instantaneous data from the final output layer's loss function. Every neuron needs a precise map of the entire network's performance. To calculate these global errors, artificial networks operate in a rigid, dual-phase training cycle. The system must completely halt its forward processing, freeze its data intake, and dedicate a separate computational phase strictly to passing errors backward. Biological brains cannot freeze. They process continuous, overlapping temporal streams of sensory data. There is no pause state to wait for an error signal to propagate backward from the motor cortex to the optic nerve. Because it requires exact weight symmetry, global error coordination, and system-wide pauses, backpropagation violates the basic physics of wetware. The core computational engine of modern artificial intelligence is biologically impossible. This creates the central tension in computational neuroscience: how to achieve deep credit assignment, the ability to train multiple hidden layers, using only forward passes and local information available right at the synapse. Leading researchers, including pioneers like Geoffrey Hinton, have attempted to resolve this by developing alternative, brain-inspired algorithms. Hinton's forward-forward algorithm is one attempt to update network weights by processing data in continuous forward passes, bypassing backpropagation entirely. To understand if these alternatives actually mimic the brain, we can evaluate 9 distinct biologically plausible algorithms, ranging from Hebbian learning to predictive coding, against a strict verification process. We evaluate them across 3 specific factors: their theoretical adherence to the physical constraints of biology, their empirical classification performance on standard datasets, and their representational similarity to actual human fMRI data. Testing the algorithms across these 3 vectors exposes a sharp structural conflict between raw algorithmic performance and true neurobiological processing. This matrix shows the 5 strict biological constraints an algorithm must satisfy to emulate physical brain tissue. A model needs asymmetric weights, local error representation, non-parallel continuous training, a reliance on discrete spiking neurons rather than continuous values, and unsigned error feedback. The local error requirement dictates that synapses must modify their connection strength based exclusively on the immediate gradient information available between 2 connecting cells, without waiting on a global loss function. Hebbian learning and spike timing dependent plasticity, or STDP, successfully fulfill this requirement. When we apply the remaining physical constraints, operating continuously without distinct backward phases, utilizing action potentials instead of floating point numbers, and processing errors without signed negative values, only Hebbian learning and STDP satisfy all 5 biological rules. But while these 2 models are theoretically flawless representations of neurobiology, they struggle to scale. Forcing an algorithm to adhere to all 5 constraints strips away the mathematical tools needed to coordinate complex, deep-layer credit assignment across millions of parameters. To maintain deep learning capabilities while avoiding biological impossibilities, researchers developed mathematical workarounds. The first major attempt is feedback alignment. Feedback alignment was designed specifically to solve the weight transport problem. Instead of using a perfectly transposed backward weight matrix to carry errors up the network, it replaces those feedback weights with a fixed, randomized matrix. The mathematical proof behind this replacement relies on a specific property. As long as the dot product of the error gradients remains positive, The random matrix still steers the synaptic updates in roughly the same direction as exact backpropagation. Direct feedback alignment iterates on this topology by establishing direct, independent connections between the global error at the output and each individual hidden layer, localizing the learning signals to bypass a sequential backward pass. By successfully routing around weight transport, feedback alignment proves that deep credit assignment is possible without exact synaptic symmetry. Predictive coding takes a different approach. Rather than finding a workaround for propagating errors backward, it abandons the traditional feed-forward then backpropagate cycle entirely. Predictive coding functions as a hierarchical generative model. High-level layers actively predict the activity of the adjacent lower-level layers. Information flows downward as a prediction, and the lower layers return the difference as a prediction error. The network's sole objective is to minimize variational free energy. The algorithm seeks to reduce the sum of the squared prediction errors at each specific layer. Backpropagation relies on global gradients. Predictive coding achieves equilibrium dynamically during the inference stage and updates its weights using purely local activity states. Both predictive coding and feedback alignment provide mathematically viable mechanisms for deep learning that respect the localized information processing constraints of physical tissue. But theoretical compliance is only the first vector. To understand how these models actually perform, we evaluate them against standard empirical image classification datasets. This scatter plot maps the algorithms based on biological plausibility and their accuracy on the CFAR 100 dataset. When tested on complex data, the strict biological models fail. Heavy in learning and STDP cannot even converge on CFAR 100. The highest performing alternative is an algorithm called local losses. It achieves near parity with traditional networks, but it only meets a single biological constraint. Local losses secures high accuracy by placing a classification layer at every step, effectively executing layer-by-layer backpropagation using global data labels. It abandons biological limits to achieve high performance. The middle ground algorithms, predictive coding and feedback alignment, demonstrate viability on simpler datasets like MNIST, but their effectiveness drops sharply on harder vision tasks. The empirical benchmarking reveals a strict inverse correlation. The more accurately an algorithm matches biological synaptic rules, the worse it performs at complex pattern classification. If biologically constrained algorithms fail at raw classification accuracy, we have to ask if they hold any actual value for modeling human cognition. To find out, we use the HAXPY dataset, a neurobiological benchmark containing fMRI data of subjects viewing distinct visual stimuli. We compare the models to this data using a representational dissimilarity matrix, or RDM. RDM is a mathematical tool that measures the internal feature geometry of an artificial model and compares it directly against human fMRI activation patterns. RDM entirely bypasses the network's final output accuracy. It looks inside the black box to measure the structural similarities of the intermediate representations the network learned while processing the image. Comparing RDM scores provides a definitive empirical test to see whether an artificial neural network is structurally processing information the way a human brain does. This chart contrasts a model's absolute accuracy against their RDM similarity score to human brains. The data reveals a core paradox. Traditional backpropagation achieves the highest classification accuracy at 79%, but yields a lower neural similarity score. Predictive coding achieves the exact opposite. Despite its lower absolute accuracy, it generates the highest structural similarity to human fMRI data. Optimizing an algorithm strictly for accuracy using global error gradients forces the network to map information in geometric patterns that are entirely alien to physical biological brains. Lower accuracy algorithms like predictive coding generate internal feature maps much closer to biological reality. Their internal geometry is organic precisely because the math is constrained by local, layer-by-layer physical limits. The current frontier in computational neuroscience is bridging this gap. Researchers must develop an algorithm that maintains local circuit-level plausibility without sacrificing computational scalability. Achieving highly advanced artificial intelligence will require moving past brute force global optimization. We must redesign our algorithms to match the physical constraints of biological wetware.